cotton, soybean, linseed, tobacco, potatoes. They all comes under complete flowers. But there are incomplete flowers like there may lack of one or more floral organs. Like if you take rice, there is absence of petals. In maize, there is absence of petals. In wheat, petals and sepals are absent. Like there are like sepals and petals are missing in these type of flowers. That is called as incomplete flowers. But again, there's another classification called as perfect flowers and imperfect flowers. Means based on the presence of male and female. If they are in the same flower, if they're having both male and female structures, then it is called as perfect flowers. Examples like rice, wheat, oat, barley, rice, sugar, cane, sorghum. Okay, like almost graminaceous, there comes under perfect flowers. And imperfect flowers, they may be having the staminate, they may having the pistil, either dioecious, like hemp, papaya, hop, and there are several examples. But first, best basic example you should remember, like papaya, hop, hemp. In perfect flowers, tobacco, sugar cane, soybean, legumes, and incomplete flowers, there are several examples. Like imperfect flowers are always incomplete. That you should remember because actually because imperfect flowers they're missing any for example if it's any missing even though it having sepal petal female definitely an incomplete flower because one of the other sex is missing in that flower, that's why it is always an incomplete. Then, what is the significance of pollination in plant breeding? It decides the gene action, it decides the reconstitution, it decides the adaptability of the particular crop, it decides the genetic purity of the crop, and it decides like how you transfer the genes, like either which method you have to follow, transfer the genes, and everything is actually determined by the mode of pollination in particular crop species. Like, what is gene action? Can anyone tell what is the definition of gene action? What are the different types of gene action present?
The ready
Hello,
to
no one is there.
dominance, epistasis.
Gene, right? In words only, gene action. Like I'm telling, like pollination is determines the gene action means. Can anyone tell how the pollination can determines the gene action?
Sir, how the genes are producing the phenotypic traits?
Yeah, how genes behave in the population. That is actually the gene action. Based on that, either gene may be additive in nature, genes may be either dominant in nature, either it may be epistatic in nature. Means how pollination is linked with gene action, how it determines. It is well known that the most of the self-pollinated crops having the additive gene action. The predominance is additive gene action. Like it is associated with the homozygotes. Additive gene action normally is associated with the homozygote. Then normally the predominance of homozygotes is present in the self-pollinated crops. In cross-pollinated crops, the predominance of dominant gene action because there will be predominance of more number of heterozygotes. Like the normal occurrence is heterozygote, then definitely the heterozygous is associated with the dominant gene action. Then definitely the mode of pollination has some link with the gene action. It determines the gene action. Okay. Anyhow, we'll discuss like what are the different types of gene action, what are the different models to explain the gene action. Can anybody tell what the different models are there to explain the gene action? Okay, we'll discuss.
The
Sir,
diallele,
generation mean analysis.
diallele,
What?
partial diallele, generation mean analysis.
Yeah,
Generation
That
mean
means
analysis.
yeah, generation mean analysis one thing. Okay. Generation may from it is like another method. Ultimately, like a single gene model and multi models are there to understand the gene action. Like either you can take mean, average, capital A, capital. I think single gene model decide M. multigenic model, single genic model. This is the homozygote and heterozygote will be there. How much your heterozygous deviating from the mid parent, based on that you can decide the gene action. Like if the heterozygote, the performance of the heterozygote is equal to the either of the homozygote, then it is called as the dominance gene action. Then if the particular heterozygous is exactly on the mid parent, then the most of the it is a like additive gene action. Based on that, like over dominance, like model and multigenic models were there to explain the gene action. And to determine the big gene action, different mating designs we use to estimate gene action and how it actually. Okay. Then if it is self-pollinated crops, there is always a predominance of additive gene action. If it is a cross-pollinated, normally the predominance of dominant gene action, that you should remember. Then the how genetic constitution was decided by the pollination, we have discussed a number of times. We will discuss in the next slide. What is genetic constitution of the particular individual? Can anybody tell?
Here in self-pollinated crops, the genetic constitution is homozygous, majorly
Yeah,
homozygous, and
homozygous.
in cross-pollinated, it's majorly heterozygous.
Heterozygous means how the allele, normally what is the zygosity of the particular genes, like alleles, it's actually genetic constitution. Then adaptability, like even the mode of pollination actually determines adaptability. You know that the cross-pollinated crops are more adaptable in nature than the self-pollinated crop. Generally, because cross-pollinated produces more recombinant, so that the better recombinant survival of the better recombinant to the immediate environment is very high. And self-pollinated also, it's like when compared to cross-pollinated crops, the adaptability to the new environment is very less. But in general, the self-pollinated are evolved from cross-pollinated crops, to that where actually after several generation of evolutions or several gradual evolution, all the mutants were eliminated. Like most of the mutants which are determinantal for this crop were eliminated, and self-pollinated are actually the very good adapters. But when it introduced to the, for example, when uncertain climate will came, like due to climate change, whatever it may be, if you want to survive in that climate, you need to change, like that the adaptability mechanisms has to take place immediately. Then in cross-pollinated, because it's amenable for cross-pollination, then there will be a production of high recombinants. If any one of the recombinant is very good to that particular environment, that will survive and it will reproduce more. Okay, it will selected by the nature. Then gradually again, it may during after several years of evolution, then definitely it may lose some character. That will be another thing. Okay, but as you remember, the cross-pollinated crops are very good for immediate adaptation than the self-pollinated crop. Then that's why the pollination actually determines the adaptability of the particular crop. Then the genetic purity, it is also matter. Like genetic purity is a very, very important point in case of seed production, because the ultimate thing in the plant breeding is seed production. Maintenance of genetic purity of the particular lines and the varieties is very, very important. The maintaining the purity in which case is much easier
Self-pollinated crops.
in self-pollinated crops, because there is no out pollination, and that's why their purity is maintained very easily when compared to cross-pollinated crops. And remember, what is the genetic purity which required for different crops? It's regularly asked in the ASR. The highest genetic purity requirement for which crop? Like which type of seeds in a seed chain? Nucleus, foundation, breeder seed, certified,
Nucleus.
nucleus and breeder seed. Okay, exactly. Then, then transfer of genes. Actually, if it is a cross-pollinated crops, then you can easily transfer the gene, just because if you allow open pollination, it will go into transfer. But if it is a self-pollinated crops, again you have to maintain some. You need some uh, pollination techniques or emasculation techniques, and you need to maintain some. Like it requires a lot of attention for transferring the genes. Okay, means type of transfer of genes, genetic purity, adaptability, genetic constitution of the plants, and the different types of gene action is actually determined by the pollination mechanism of a particular crop. Actually, it is a nearly 10 marks question in the AS lecture. AS actually, like how the pollen, what is the significance of pollination in plant breeding? Direct question. Then, what is the genetic structure of the population? Like if it is a pure line, it's highly self-bonded. It is normally followed in highly self-bonded crops because the definition of pure line itself is says it is normally in self-bonded crop. It is a single homozygote and it's homogeneous in nature. When you take the hybrid, it is cross-pollinated. It also produces like we can also produce hybrid in self-bonded crops. It is single zygote and homogeneous in nature. And rest other thing already know the multi-line it's self-bonded crops and it's a mixture of several isogenic homozygotes and it's a heterogeneous in nature. And where in the case multilines are mostly used for which resistance? Normally for the biotic resistance, multilines are widely used. Then varietal blends, it's just a mixture of all your self-pollinated or it can be followed in cross-pollinated crops, but it is highly heterogeneous in nature and several homozygotes are there. And you can also do varietal blends in cross-pollinated crops, but maintenance is little difficult. Then synthetics and composites is highly heterozygous in nature and heterogeneous in nature. We have discussed in detail how to develop the like except multilines. I have discussed everything. Like what is pure line, what is hybrid, what is composite, what is synthetics. Please refer my earlier classes for that. Then in cross-pollinated crops, general they are highly heterozygous in nature without exception. Like if you enforce any inbreeding, then definitely there will be deterioration in the general vigor and other adverse effect. What is the term is called as inbreeding depression. We have discussed in detail. Then what is the like heterozygosity is a very essential feature of any commercial varieties of the cross-pollinated cross because why any term used a number of times the term where like heterozygosity is the cross-pollinated crops having more favor in when they are present in the heterozygous condition. What is the term?
Heterosis.
Heterozygotic balance.
Heterozygous balance. Okay. Then, the self-pollinated species, there may be mixture of many closely related homozygous, and they're vigorous in normally in homozygotic condition. Normally, that's why we every breeding method is aimed to develop the pure line. We discussed that's why. Like, what are the different mechanisms that promotes the self-pollination is hermaprodism, like homozygamy, cleistinogamy. Normally, hermaphrodite plants having both androecium and gynoecium present in the same flower. We had like example of perfect flower. Then you know that many rice is the hermaphrodite. Then examples it's a very basic. Then homogamy is the one of the important mechanism that actually promotes your self-pollination. Means homogamy means what? Like it's just a maturation of male and female same time. And the same like another word like synchrony that we have discussed in detail while discussing about hybrid seed production. Then another one of the important mechanisms that promotes the self-pollination is cleistinogamy. And even though it is basic, like many times these are the definition of fixed questions. Either homogamy, cleistinogamy, hermaphrodism, like one of the questions definitely will come in the ASB exam. Then the any example like chickpea, wood bean, mung bean. Actually, one trick they will follow in while asking this type of question, they will give some a rare species. Like they will ask some melon crops, either it's a cleistinogamous or monoecious or homogamous. They'll ask like this. They will give an example and which method is promoting the self-pollination. They'll directly give. For example, in last year ASB, they given a tomato and which mechanism which actually promotes the self-pollination in tomato. Can anybody tell which mechanism actually promotes self-pollination in tomato?
Hermaphrodite, sir.
Yeah, hermaphrodite is one mechanism. Another mechanism?
Homogamy, I think, sir.
Yeah, homogamy. In tomato, they actually you didn't think there is all options were not there. There are different options. Any other mechanism other than this? Even hermaphrodism is there. If there is a diagamy in nature, like diagamy is present, then definitely there will be a out pollination. In homogamy also there, but um, even after receptivity, like even the stigma is receptive for longer time, then definitely there may be open pollination. Okay. Then clestonogram, actually it's the most important method of actually if the plant is clestonogamous, it's never open, like never cross pollinate because it's completely closed. Like anthers and stigma, it's completely closed by keel petal. Then definitely there will be no out pollination. Only after if you make any changes by in artificial means that you can make plant is like cross pollination, but naturally it will never cross pollinate if it is a clestonogamous in nature. But casmogamy, there will be a mechanisms like because after fertilization actually flower opens, but even there is little bit chance of occurrence of cross pollination present if it's a casmogamy, but rare. But clestonomy and casmogamy is actually very important mechanism that actually for self pollination, like graminaceae like wheat, barley, oats, lettuce. And in tomato, you, you see the structure of the tomato, like flower structure of the tomato. The anthers are surrounded, like sorry, the stigma is actually surrounded immediately by the anthers. Like anthers will close the stigma. Means the pollen produces in anther definitely pollinates the stigma. Even though the flower, it's the petals which not covers the stigma and the anther. Although the structure of anther surrounding the stigma, it's like that. It enhances the self. Actually, it was directly asked this mechanism in tomato. It actually promotes the self pollination. There are different examples for different crops. Please read it actually because there are in BDC it's mentioned in detail. Please read it. Then a genetic what are the structure like what are the things which promotes cross pollination? I actually are not mentioned here. Yeah, we have trichogamy, protogynous condition, protoandrous condition, heterostyle, unisexuality of the flower, incompatibility, male sterility, and somatoplastic sterility. There are different mechanisms. You know that what is dichogamy? Means maturation of anther and stigma at different times, which normally discourage self-fertilization, and protogynous, which example is permulate and the walnut. We all remember the permulate for the protogynous, and walnut is also actually in some varieties, in some like varieties of walnut, that is protogynous in nature. There is another example, it's asked in I think several IRA entrance exam previously. Actually, soybean is also soybean also a protogynous in some nature actually. In some varieties, it's not like all the varieties. There are several exceptions in there. In some varieties of the soybean, there is a predominance of protogynous in nature, like predominance of protogynous condition. Okay, that you should remember. In exam, we know that these concepts are very easier, but sometimes they will ask rare examples so that we will get confused. Like we'll get confused. That's why remembering the example in these type of questions is very important. Like walnut, it's a protogynous in nature. Pearl millet is also protogynous in nature, and the protoandry, it is also several examples. Normally, we remember maize only the example, but even sugar beet also protoandrous in nature. Carrot is also several species, several cultivars of and several species of carrot is also protoandrous in nature. And heterostyle, many times it asked in the examples. Remember example. What are the different example for heterostyle? The famous example of heterostyle. Who knows?
Linseed.
Primula. Primula, sir.
Yeah, Primula. One of the famous examples. Primula was like long style, medium length style, short style. Okay. Remember those examples. And in brinjal also several some sort of heterostyle present. Yeah, other classical examples are there. Please refer that. What are the different types of heterostyle? Like while reading the several seed technology book, NC single book, it's very good book. Please refer that. Okay. Several examples are given, and it's very important to remember for the part B purpose. Okay.
Sir, which book,
Then
sir?
NC single book by Seed Science and Technology. NC single. I will share. I think I will share that name. NC single. NC Sing something is there. Seed Science and Technology by NC Single. At least read some chapters like mode of pollination, mechanism of pollination, and seed like different seed dispersal mechanisms. It's nicely explained actually. Like different and many times even I also made mistakes many times. Even though I remember many examples, they'll ask different examples like the pollinator species, like a pollinator, like pollinator of if it is pollinated by bee, what is it called as? Different like sarcophily, anemophily, entomophily. There are different phyllis. I don't know how many phyllis are there. Every year there will be a new question, and every year we make mistakes. Like, but nobody will attend if they ask very difficult questions. But at least try to remember. And in that book, it's very like different pollinator mechanisms, like different like hydrophily. What is the examples? Which crop the hydrophily was predominant, and pollination by snakes, pollination by ants, pollination by mites. Last time there, what is the if pollination by mites? What is that term is called as? If hymenopteran species do pollination, what is the mode of pollination? If bats do pollination, what is the type of pollination? Mode of pollination called as? If ants do pollination, if it is molluscans do pollination, what is the mechanism of
Myrmecophily and
So for
then
bats, it is chariotophily.
Erythrophily. Myophily means what? Transmitted by like pollinated by myophily.
Flies, sir.
Yes, hymenopteran species. It is pollination by there are several like reptiles means the pollination by reptiles. It asked in 2022. Every year, 2022, 23, 24, every year they ask different exam. This year they were asked, I think,
Saprophilia.
bumblebee. I think I don't. Yeah, superphily bumblebee. This year I actually referred, but this year they were asked like uh, I think the bumblebee pollination by bumblebee. What is it called as? Several examples you should remember like, and so was several botany books like sorry, read some important botany book called as which is book like forgotten the economic botany by anyone remember the author name? Economic botany.
Kochar,
Will
sir.
just
Kochar.
refer any economic botany.
So that
Kochar book
Amit Kumar.
actually. Kochar and Amit Kumar. In Amit Kumar, it is not that much in detail, but you refer Kochar book. Okay. Then examples are very very important. That's why I wanted I just kept these slides. And what is somatoplastic sterility? It actually predominance in lucerne. Okay, embryo resulting from the cross fertilization grow more rapidly than the embryos of the self fertilization. So that all your self fertilization, the embryos which are formed from self fertilization, it got eliminated because it's the embryos which are more vigorous when it developed by the when it is received the pollination from pollens from the different crops. Okay, if it is like a kind of male sterility, like self incompatibility, but it's a, even though fertilization occurs, both what is the difference between incompatibility and somatoplasmic sterility is here the fertilization occurs, but the growth of the embryo is very very vigorous when it is received across pollen, like the pollen from the other plants. But if it is received pollen from the same plant, the growth of the embryo is restricted. There's the difference between somatoplasmic sterility and incompatibility. And we'll discuss what is the male sterility, different male sterility mechanisms, and it promotes the cross-pollinated. And you know that what is unisexuality occurs, like if there are many examples, either it may be dioecious in nature or monoecious in nature. And remember, there are different examples they will going to ask for the monoecious. Like they will ask several medicinal plants, several like non-economic plants. Definitely there will be a question which you can't guess also. Like They'll give date. There will species of date palm. They'll give some species of walnuts. They'll species of some fiber crops. Then they will ask what is monoecious, either it may be dioecious. Okay, like chow chow is cross pollinated or self pollinated. Chow chow,
Cross, I think so.
the highly cross pollinated. Okay, why like some unpredictable like examples they'll give and they'll ask which is the mode of pollination, which is the mechanism which that promotes self or cross pollination, that you should be very uh, stringent in that one while reading those concepts. Okay, don't neglect it. Then, and remember what is somatoplastic sterility, what is the difference between incompatibility and somatoplastic sterility, what is male sterility, and different examples, very important. Then, like as I just discussed, for successful hybrid seed production, we need a very cost-effective emasculation techniques. That is emasculation techniques. There are several methods of emasculation techniques that we follow, like hand emasculation. It's kind like some hand emasculation, very much easier in cotton and tomato. That's why still now the people are using hand emasculation. In some crops, genetic emasculation is very important. For take the rice, any other highly self-potted crops. In that case, like where flower structure itself is a very small, where hand pollination is not that amenable, that's not economical. Okay, main determinant is economic uh, points, like an economic factor of producing the hybrid seed production at reasonable cost. It's a very important determining which emasculation method they are using in that particular crop. It's not like that always a genetic emasculation is very much cost effective in all the crops. You can't say that. Okay, even some using like developing rather than developing the genetic emasculation lines, like different cell lines, just detasseling and pollinate. It's very easy when compared to developing these methods in maize. In cotton also, dusting and pollination is very easy when compared to developing these systems. That's why it's like it depends on the economical point of view, like which method is more easier and most uh, cheaper to do emasculation and do produce the hybrid seed. Then there are different genetic emasculation methods like male sterility, either you can use special condition that present in capture, either you can use self-incompatibility that present. Normally now also we are using this system in brassica, and in male sterility it's regularly used in different rice, maize, and it is used in sunflower. And which is the type of self-incompatibility that present in the mango? Can anyone guess? Porophytic. It asks in sorry exam ASRB actually. Sporophytic self-incompatibility that present in the maize. And in brassica, which type of self-incompatibility present?
Gametophytics.
Gametophytic. Is it right? In solanaceous, it's I think solanaceous gametophytic and sporophytic is in brassica, I think. Once check. Okay. Then and another method is chemical emasculation. We'll discuss in detail, don't worry. And self-incompatibility, I'm going to discuss tomorrow. But we'll discuss all male sterility mechanisms today. Then what is male sterility? Male sterility is means what is male sterility? Is defined. In a flower, where female
Yeah.
gamete is normal, male gamete is abnormal. Okay, male gamete is infertile. Okay, that is a simple definition. There are different types of male sterility. Can anyone explain? Please enlist.
Cytoplasmic male sterility, genetic male sterility. Yeah. And CGMS.
Okay, CGMS. Okay. Then another types based on the functional male sterility, structural male sterility. Remember all the point. What is functional male sterility? What is structural male sterility? Okay, what is another type? I'm not getting. Okay. Remember all the things. Okay. The majorly genetic male sterility, cytoplasmic genetic male sterility, and In that, again, another one is CGMS system, cytoplasmic genetic male sterility. We will discuss one by one in detail. The genetic male sterility, the male sterility is caused by the genes which are present in the nucleus. Cytoplasmic male sterility is the male sterility caused by the genes which present in the cytoplasm. Either it is in mitochondria, either chloroplast. Generally, till now today, we identified the genes which are present only in mitochondria that causes male sterility in the plants. Okay, and genetic male sterility which follow typical Mendelian ratios that follow a Mendelian inheritance, but cytoplasmic is a non-Mendelian inheritance. So because it's a cytoplasmic inheritance mechanism. Then genetic instability, there are again several classification, that is environmental insensitive male sterility, environmental sensitive male sterility, another one is transgenic male sterility. Means that the expression of the male sterility which is not affected by the environment, and in this environmental sensitive male sterility, expression of male sterility which depends on the where it is growing. Like environmental factors are very much determinantal, deciding either the gametes will be fertile or infertile. Then transgenic male sterility is male sterility is actually was created by introducing the gene construct which are responsible for inducing male sterility. We'll discuss all the types in detail. Then when it comes to genetic male sterility, which is insensitive to the environment, where actually it is governed by many times, it is governed by a single recessive gene. There are several examples which are male sterility, which is also governed by a capital allele of the particular gene. Either it may be recessive allele, majority of the times it's governed by a single recessive allele. Then it is also governed by the dominant allele also in several crops. For example, like some pea crop. Okay, there are several examples are noted. And majoritarily it is governed by a single recessive gene, a single recessive allele. Then when you're looking to the inheritance pattern of this genetic male sterility, for example, this when there is genotype having the homozygous recessive condition, the plant will be male sterile. If it is dominant, particular gene is particular allele of this gene is dominant, the plant will be fertile. When you make the cross, when a heterozygous condition will be fertile. Again, if you self it, there will be 1:2, the segregation of the male sterile gene into 1:2:1. Again, 1/4 of this particular F2 population will be male steriles. So to maintain this, for maintenance, you just cross male sterile lines with the maintainer line, which is heterozygous for the particular gene, male sterile gene. Then you are getting 1:1 segregation. In the field, you have to eliminate percent of the plant because you are getting 50% male fertile and male sterile plants. Then you should be remove the removal rowing should be before they shed the pollen. If they shed the pollen, everything will go to spoil. That's why and this method is not that much commonly used because of this limitation. Because you have to maintain a double the population to maintain this particular male fertile male sterile line. This is one of the major disadvantages of genetic male sterility. Then what are the different and stages that, like in tomato there are examples at different stages different male sterile gene will activate and will lead to male sterility. For example, MS2 and MS3 gene is a it occurs like it causes sterility when there is a premeiotic stage, zygote, diploid, prophase like MSK3 gene, tetrad, MS6 and MS31 gene, and either it may be MS5 and MS8. These are all actually identified in tomato. I don't remember just for purpose. And I have taken all this information from the medicine book and medicine classes. Okay. Then what are the environmental genetic study? There again it is classified into two systems like temperature sensitive male sterility and reverse temperature sensitive male sterility. And please remember the examples actually many times it asked in the exam. Remember the example at least in rice. Yesterday we have discussed, but I forgot to give the examples. Like for example, in a PA64S rice, majorly the temperature sensitive male sterility is predominant, and in case of J2007S, it's one type of cultivar where the reverse temperature sensitive male sterility is predominant. Sorry, like temperature sensitive male sterility means, for example, if it is crossed to critical temperature, it's above the critical temperature. Like in this particular line, 23.5 is the critical temperature. The temperature above this temperature, then definitely there will be a male sterility. But it's not like that always above. There may be temperatures less than this particular critical temperature. There may be sterility. Like that's why temperature sensitive male sterility and reverse temperature sensitive male sterility. Again, there is another classification called as photoperiod sensitive genetic male sterility. Again, there is a classification photoperiod male sterility and reverse photoperiod sensitive male sterility. It means that, for example, you take the NK50 58S rice, where if it is a long, the photoperiod length is more than it is 13 hours 45 minutes, the plant will become sterile. Also, it is not like so. Even some genotypes, if it is the photoperiod length shorter than the uh, hello, slides are visible.
No, sir.
Actually, slides are visible now. Actually, I got disconnected
No,
in
sir.
middle.
No, sir. I
Okay,
think
okay.
your network is weak,
Yeah, yeah.
sir. That's why your voice is also
Yeah,
breaking.
I think.
I thought my network
Okay,
is
okay.
weak.
No, no, my network is not good actually. In last 2 3 days, it is like. Please, whenever you are facing problem, please mention. Okay, because I don't know what happening there.
Okay, sir.
Now it is clear. Was
Yes, sir, it is clear.
yeah. Then photoperiod sensitive genetic male sterility. And please remember it. It's not like many times we have a concept like we decide like okay if temperature when it is crosses above that particular critical temperature sterility will occur. It's not like that always. Maybe if the temperature below that also maybe sterility will going to occur. Like in case of J27S. Okay, that is called as reverse temperature sensitive male sterility. Okay. Then in the similar way, photoperiod also. If it is crosses the particular critical photoperiod length, definitely there will be occurrence of the male sterility. And it's also if it is lesser than that of the like if it is shorter than that of that particular threshold, it may also lead to sterility in some genotypes. It's not like that. It's always above or always below that of the particular critical temperature. That you should remember. Then there are several lines like we already discussed, like non-Ken 58S, MG201, and there are different example. It's actually in 1973. It's spontaneous mutation. If the male sterility arises due to spontaneous mutation, like we have discussed in detail. Then there are example. There is no need of remembering all the examples. Just remember the TGMS and PGMS systems are more predominant in case of rice. That too in China. That's why hybrid rice is predominant in China. Then when it comes to hybrid seed production by using this system, as I earlier yesterday we had discussed in detail, although I will revise. Then, for example, you want to maintain that particular sterile line, just grow in a, for example, take location A, location B, and the particular line which are you are using, for example, take 5460S line, which is in location A, it is normally fertile. The gametes are fertile, the males are fertile. But if you bring this 5460 variety into the location B, then definitely it will be going to be sterile. Where you can do hybrid seed production, where the sterility will occur, there only you can do hybrid seed production. In this case, in location B is very much suitable for the hybrid seed production, and location A is suitable for the multiplication, maintenance. Like whenever you want to maintain that particular line, you just bring this 5460 into location A, then your the maintenance of like multiplication or maintenance of this TGMS line will be happen. Then if it is in location B, it will be a male sterile. You can cross with any line if you want to cross, like any inbred favorable inbred that you have chosen already. Then you do hybrid seed production you just make crosses, then you produce hybrid, then you can going for direct release, testing and release. Location B is favorable for hybrid seed production, and location A is favorable for maintenance of your line. Then here there is an elimination of your maintainer line. There is no need of nursery, or there is no maintenance of B line, like maintainer line. There is one of the greatest advantage of TGMS systems over the three line method of breeding. Then there is another predominant technology actually is published in 2016, seed production technology, where the maintainer line is transgenic, but the ultimate product is non-transgenic. It's like transgenic based non-transgenic male sterility. It actually came in first paper published in 2016 by Yangzhou et al. Actually, most of the things which are occurred in China only, published in very important journal called as Plant Biotechnology Journal. Then what is this seed production technology means? Here we are creating, like we are introducing in the male sterile plant, we are introducing one transient construct that having the counterpart allele of the male sterile gene, that is the dominant male sterile allele. Then PK gene, which is pollen killer gene. Like normally we are using ZM, SS, like zygotic lethal genes, like PK pollen killer gene, and one is one marker gene, like it produces the red fluorescence when it is in the heterozygous condition, like it is in a dominant condition. This gene construct actually introduced, for example, into the male sterile plant. Then, if it is crossed with the male sterile plant, like MSMS, then it produces both male sterile plant and male sterile plant with this particular gene, and also with the male fertile plants with the particular genes. Then, how to eliminate, for example, the male sterile plant, if it is producing that color, means you just directly eliminate. Then here that particular plant is used for hybrid seed production. Here also male sterile plant is there, but it is a transgenic in nature, because like just discard, because sorry, this is also male sterile plant, but you don't have any color. That the elimination is very difficult. That's why you eliminate this also. Then another maintainer line again you are going to get. Then you recycle for next cycle of maintenance of the male sterile plant. Understood what is seed production technology? Either you read this paper, like in detail explanation how, like fluorescence with the nice photographs also given, how to identify, how to sort the colored individuals, how to sort non-colored individuals. Okay, just to introduce this construct, into the male sterile plant, then it's crossed with male sterile plant, then it also selfed, then again segregation will occur. Here, how to eliminate the fertile plants? Wherever you are getting the color, based on the color, you just eliminate that plant and allow only male sterile plant for the use, that allow only male sterile plant to grow, then use it for hybrid. After selfing this plant also it produces male sterile plant, but no use. And this again, which is act as a maintainer line, then use it for recycling. Understood this method? What is seed production technology?
Sir, it is little bit same as normal maintainer line, no, sir?
Yeah, it is same as, but here you are using particular gene, DS red, where it is like it produces red color. Based on the red color plants, like where after like you just harvest the seed, based on the color sorting by color sorter, you can eliminate all your that particular transgenic plants, and that are fertile. Wherever your color will come, that it is fertile plant. Okay, you just remove all your fertile plant, you just maintain only sterile plant. That's the only crux. It is like genetic male sterility only, but here the elimination of male sterile plant is very very difficult. So male fertile plants is very very difficult, because in field you have to take the pollen, then you have to check the pollen viability test, either it is sterile or fertile. It's very tedious task. Based on the color of the particular seed, you can just maintain that, just allow everything to pollinate and fertilize. After that, based on the color of the particular seed, you just eliminate all fertile plant, and you keep only sterile plant. That is the only use of this SPT technology. Understood?
Yes, sir.
It's same as like genetic maturity, but your elimination of fertile plants is based on the color sorting mechanism. This particular marker gene, which produces the color, based on that, you can eliminate all your fertile plant. One thing you just remember in this, like it uses a transgenic, like Barnes and bar system. Ultimate product is you are getting a transgenic, but you are using a transgenic only to maintain the moisture of plant. But while you are eliminating all your colored plants, so that in that only transgene was present. In non-colored plant, transgene was not going to present. That is the only, like ultimate product is non-transgenic, but while maintaining in seed production plot, it is transgenic. But ultimate product is non-transgenic. That is one of the greatest advantage of this technology. But it is not used by anyone. But it's still recently, like 10 years back, it's published. 9 years back, it's published. But still, it is in like a trial. Maybe no varieties are released by using this system. Okay. Just for you, just whenever like any technology which uses transgenic moisture line and produces non-transgenic plants, that is called as SPT technology. Remember that point. And which construct they are using? They are using pollen killer gene, the construct which having the dominant male sterile allele and pollen killer gene, and also DS red gene, which use it for the elimination of your all male fertile plants and transgenic. Okay. Then other method is transgenic male sterility is Barnes and Barster method. It's a transgenic male sterility, but this is non-transgenic male sterility. Even though it uses transgenic construct only for maintenance, but the final product is not transgenic. But in this case, Barnes and Barster technology, your final product is a transgenic. That's why still the varieties like a hybrid which are released by using this Barnes and Barster mechanism still in the court. Okay. Still, they are not regulated to use this particular hybrids for the commercial cultivation because it is a transgenic. It has to follow the transgenic rules of 1986, like 1989. Then, the what is Barnes and Barster system? This regularly asked question. It's developed by whom? Scientists who actually developed this Barnes and Barster system in India, in mustard. I'm asking in India. Deepak Pental. Okay, remember the name. Deepak Pental is from South Delhi University, like University of Delhi South Campus. Actually, he developed Barnes and Barcer system, and the variety actually the release now it is cleared for environmental release. That is called as DMH11. Remember the example DMH11, and also remember the parent actually. Husa bold crossed with some other variety. I forgotten. Like DMH11 actually developed by Deepak Pental by using the Barnes and Barster male sterility system. Normally, where it is same as like genetic male sterility, but the only case is how you introduce the male sterility by transgene. Like if the Barnes gene is used, it is creates the male sterility. Barster it acts like a restorer and the maintainer gene, like just if you along with the Barnes gene, you just put one gene called as bar gene. It is sensitive to the herbicide. Those containing that gene, they definitely they'll be resistant to the phosphonaithricin herbicide. If there is a bar gene was not present, definitely the plants will be sensitive to the this broad spectrum herbicide. Then based on the spraying this herbicide, you can eliminate all your fertile plants. In detail, we will discuss. For example, if you are introducing Barnes construct into the plant, then plant will become male sterile. If you are introducing nothing, just it's a maintenance line. Then you are crossing normal line with this Barnes transgenic line. Then you are producing a 50-50%. 50% Barnes line and 50% normal line. This normal line don't have any bar gene. Like bar gene is actually gene which are confers the resistance for the phosphonaithricin herbicide. If the plants which are not containing a transgene along with the bar gene, they are sensitive to the phosphonaithricin. When you are spraying the herbicide, your all fertile plants will get eliminated. Only transgenic plants will be remained. But here, this is transgenic. This is used for the hybrid seed production. That's why it is not released. But SPT technology, Here, this particular plant is non-transgenic that used for hybrid seed production. This is non-transgenic. Okay, that is the only difference between Barnes system and your SPT technology. But here, the plant which is male sterilite is a highly transgenic in nature because it includes the construct of your bar gene along with Barnes gene. Then, when it is crossed with the line which having the construct of Barster actually it acts as a restorer gene. Restorer means whenever the Barnes comes along with the barster will suppress the activity of the Barnes. The Barnes actually produces the RNA which are determinantal for the reticulocyte development. But the barster, what it will do, it will degrade this particular RNA produced by the Barnes. That's why the barster act as the restorer gene for the Barnes male sterility. That's why barster used as a restorer line. When you cross the male sterile line with the restorer line, then definitely the fertility is restored in hybrid plants that can be used for your commercial cultivation. Understood what is Barster and Barnes mechanism of male sterility and who developed
That
it?
phosphoinothricin spray, for that the insensitivity is caused by bar gene, sir.
Bar gene, not Barnes. See, in gene construct having in that case gene construct having Barnes gene and bar gene. Two genes will be there. Wherever the bar gene is there, it is resistant to herbicide. Means your Barnes gene is actually linked with your bar gene, and this particular construct introduced into male sterile plant. Sorry, like brassica plant, then plant will become male sterile. If this gene construct is not introduced in the particular plant, then plant will not become male sterile. Why this system is used? Because the male sterility system which present in this brassica, like maybe there is a high percent of self pollination, there is a low frequency of fertility, like sterility. Then there is several problem in natural system. That's why there is a development of the very efficient system called as Barnes and Barser system. Okay. Whenever the bar gene is there, then definitely this bar gene actually highly susceptible, like highly resistant to phosphoinothricin. There are several
So
genes. Okay.
these genes are isolated from some bacteria, na, sir.
Yeah. Barnes gene, bar gene, all they are from different bacteria like Pseudomonas like aminofolium, something Pseudomonas aminofolium. Okay, they're all from the bacterial origin. That's why it's a transgenic. Barnes, Barster, Bargene, all from the bacterial origin. They are actually the gene construct. These are gene from the bacterial construct. Gene isolated from the bacteria and they are made into the construct. This construct introduced into the DNA by recombinant DNA technology. Then they are cloned, they are regenerated. The transgenic plants used for the this one seed production. Understood? First, you have to develop the
Yes, sir.
lines actually. First, there will be normal line, then isolate the cells, then by agrobacterium tuminifaciens, you introduce your transgene construct into the your normal line cells, then regenerate the plant, then develop the transgene plant, then confirm the homozygosity for the particular genes. Then after that, you use these lines for the hybrid like hybrid seed production. Understood?
Yes, sir.
Yeah. Then there are several systems. It's again very important. Many times, definitely there will be 2 to 3 questions from this part, like male sterility, reproductive mechanisms, examples. Okay, like in Brassica, there will be CMS oguru cytoplasm. It is from the Japanese wild radish, and it's mostly used actually. There are several hybrids are produced by using this CMS oguru cytoplasm because it is a non-transgenic. It's like the varieties which are developed by this male sterility it's a normal conventional hybrids. That's why it's majority of the hybrids which released in the brassica by using CMS robust cytoplasm. But nowadays it's a because of chlorosis, because of the genes which are present in the cytoplasm, which leads to chlorosis that is reducing the yield. Again, we'll discuss how to maintain that in next class. Then sunflower, there will be PET cytoplasm. It is from the Helianthus petiolaris, which is commercially used. In maize, there are different cytoplasms: CMSC, CMSS, CMST, and most commonly used is CMST. But nowadays nobody is using because of it's highly susceptible for the southern codleaf blight caused by Bipolaris madis, which caused epidemics in 1970s in USA. In rice, the wild abortive is majorly used. It's from the Oryza rufipogam and CMS BT from Oryza sativa, and Triticum aestivum majorly used. Your this one is Triticum timophobi. What is the genetic constitution of like genome of Triticum timophobi? Anybody remember?
Double A, double G.
Double A, double G. Okay, directly in the exam. Okay, directly the male genome of system of wheat. You should know what is the male sterility line, the male sterility from which source become timophoby. Then you should remember the genome of this particular timophoby. Okay.
Sir,
Then there is
after
again some
developing,
restored. Hello.
sir, after developing hybrids only, we will introduce this cytoplasmic genes. Yeah, or we can introduce it during development of the hybrid also, sir, due to the hybrid program.
Now, first you will decide the parents. Actually, first you will decide the parents. First you will decide the parents. Actually, then after that we will bring this male sterility system those parents for the commercial seed production. If
Okay,
it
sir.
depends actually, if there is already male sterile lines are there, then directly you can use some. There are several repository of all male sterile inbreds line and female like lines are there, then directly use. If the no it is not present, first you decide inbred lines, then you introduce male sterility system to that inbred lines, then you do hybrid seed production. Understood?
Okay, sir. Yes, sir.
First you have to decide the inbreds, then select the hybrid combination. Like first, how to choose the parents I have discussed. Based on several evaluation methods, you have to decide which inbred combination is giving better heterosis. Then after that, for commercial seed production, you introduce male sterility systems in it. Then you do for hybrid seed production. Understood? First, you have to decide the inbreds. Then later we introduce.
Okay, sir.
If it's already the introduced systems are there, then definitely use that. It depends. But normally, first you have to identify the inbreds, then you bring the male sterility systems to that inbred, then you do hybrid seed production.
Sir,
Okay?
hello, sir.
Yeah,
Sir,
yeah.
if we
We
means
normally actually we make large number of crosses, like several we developed several restorer lines in different backgrounds, and we will introduce male sterility system like in different backgrounds. Then we use lot of crosses by using different mating designs, and we will decide the best hybrids. Then we will go for direct hybrid seed production. That also there. Need something you are telling?
yes, sir. Same thing I was telling because if we introduce maize after introduction, then again we have to check for combining ability. So if we
Yeah, yeah.
initially we
No,
introduce
I'm not
CMS
telling.
and
No, I'm not telling after developing hybrid directly introduce the sterility system in hybrid. I'm not telling it. While choosing the parent, either while choosing the parents or after choosing the parents, you can introduce. But again, decide like you should check the combining ability for restorer lines. Okay, then because either you develop the inbreds, different inbreds by introducing these restorer genes and the male sterile genes. Then you make all your combining ability test and all those things, you can develop hybrid. So that's done. Yeah. Now you clear?
yes, sir.
Yeah. There are several papers like see R genes, like introduce R genes and making heterotic combination. There are several papers you can read. Okay? Then heterotic pooling, how we'll do like based on this identification of restorer genes itself is a tedious task. Like first they will create all the restorer lines in different background. Then they will create the mesh filter system lines in all the backgrounds. Then they will use those lines, R line, A line, and they'll making heterotic crosses. Then they will decide which hybrid is better hybrid. In Parmalat, actually one paper was there. You just read that. Like A line, R line, there will be several they will maintain this whole population. Several lines designated A line, several designated R line. They will they'll develop all these A lines, R lines in different backgrounds. Then they will do all these particular crosses in different mating designs. They'll evaluate that crosses which is performing better hybrid, then directly they will release that. This can be done. Now you understood how actually it will takes place. At breeding nursery,
Okay.
there will be a lot of things. In pearl millet and maize, it's a common thing because then definitely they are using actually pearl millet, majorly in India, pearl millet and another crop is sorghum. All the A line, they maintain A lines, R lines in several backgrounds, so they are already developed. They just keep on testing the lines, different A line and R line, A line, B line. The best combination, if they get best combination, then directly they will release. It's like a regular breeding program. Like in pearl millet and sorghum, it's a regular program actually. They are continuously doing the same thing, and they developed these R lines and A lines in several backgrounds. Okay. Then there are that's what I'm discussing. Like there are different restorer genes like RFO and RFK1. Please remember this example. Writing example is very important in CMS OGU and in rice like like RF3 and RF4. Please remember this designation also because in qualifying, like for me, qualifying they ask which are the restorer genes which are present in the uh, rice. I just told R genes, directly R genes, but I am not remembering exactly RF3 and RF4. Okay, that's what very important while writing the examples, especially in some crops. It's very important. You remember all these examples is present in the medicine book. Okay. This is what I want to discuss about male sterility. Any doubts regarding it?
No.
Sir, I have one question, sir.
Yeah.
Sir, what will be the gene action present in inbred lines?
Inbred lines. Actually, it will be normally inbred lines are tested, developed by evaluating for GCA. Normally, there will additive gene action will be predominant, but it also tested for SCA also after that. Like in several inbred combination, then definitely they will be tested for a particular combination. There will be high at particular combination. Combination there will be high SCA. Then based on that, we will choose the hybrid combination. But first, the development of inbred based on the GCA test, then definitely there will be a additive gene action. Then after that, we
Sir,
test the better inbred. Then there will be which having the good SCA. Then we will select the type.
yes, sir. Sir, if there is a dominant gene action in inbreds, then the dominant gene action can't be fixed, right, sir? So, but the
No.
performance will be,
That cannot be fixed. No, that cannot be fixed.
sir.
There will be always a question. Like normally we see that hybrids, they will ask in hybrids, there is no presence of additive gene action. It's wrong actually. There will always be presence of additive gene action. The presence of additive gene action, we can't say that it seizes the dominant gene action. Okay.
Sir, no, sir. What I was saying means, so only additive gene action can be fixed, right?
Yeah, it is fixed.
Sir, but
Fixed
when we
and
take inbreds, sir,
yeah. Yeah.
sir, but we take inbreds, so the performance will not means after some generation, will inbreds show
Yeah.
same performance, sir? Means after
No.
6 or 7 cycles of inbreds, will inbreds show same performance?
No. Actually, yeah, it will not show that same performance. First, we will do early generation testing. Then, after several generation of selfing, we are again and each and generation we are evaluating, and we are taking care that we are selecting the best performing lines. Okay, then definitely if you do any selection, if you don't do any selection, definitely the determinantal allele will keep on increasing, the frequencies keep on increasing, then definitely you are going to get a reduced performance in the inbred if it is a highly cross-pollinated. But normally what we will do after early testing also, after top cross testing, after developing a superior inbred line, we evaluate at every generation, then we will select the better one. That highly vigorous lines only we will select. Then we keep on increasing that particular in like we are only advancing them only. It indirectly increases the which are alleles which are good for the particular inbred. The frequency is only going to increase which is favorable to the particular inbred. Normally, that we actually do the selection that actually determines. If you don't do any selection, definitely it will affect the performance of that inbred. Understood?
Yes, sir.
That's why we do selection. Earlier, why the hybrids are, even though single cross hybrids are came, but at some point of time, it's not become very famous. Why? Because this is the one of the disadvantage. Because the good inbreds are not available. Why good inbreds are not available? There is no techniques to isolate the better inbreds. After like GCA test came, early testing came, and then top cross testing came, and biometrical techniques are developed, biometric systems came. Then the statistics came, like Fisher given several experimental designs to eliminate the environmental effects, genetic effects. Based on all the integration of all these things, we can able to see the particular the plant which is showing high vigorous than the less vigorous plant in a very clear way. So that the selection efficiency is increases, then the availability of the good inbreds will be more in that time. Then again, single cross hybrid game taken boom actually. That's what the point.
Sir,
Now you understand?
means, if we
Yeah, you
consider
can
a
ask.
hypothesis situation, sir, if we consider hypothesis situation where no detrimental alleles are not present in the inbred population, so
Yeah,
will again the performance decrease in those inbreds?
there is no see why inbreeding depression will happen. What is
So
the
no,
genetic
sir.
mechanism of inbreeding depression?
So, sir, that's why I told if you consider hypothesis situation, again will it decrease,
Then definitely
sir?
there is no reduction in the performance.
Sir,
If everything is
then,
good, why it will?
sir, then the dominant gene action means if the plant is there, then some of the traits will show additive gene action and some additive will show dominant gene action. So if performance is not
Yeah.
decreasing, it doesn't mean that we have fixed the dominant gene action.
No, actually sometimes no. How can you say that dominant gene action will always give a better performance and additive gene
Sir,
will always give a better performance?
no, sir, I'm not telling about better performance. I'm just telling that if performance is not decreased over the
Yeah.
years, then it doesn't mean that gene action is fixed.
It doesn't mean that gene action is fixed.
Yes, sir, means what again I will repeat my question, sir. If we consider hypothesis
Yeah.
situation where no detrimental
Yeah.
alleles are not present in inbred population, then will
Yeah.
the performance will decrease over the years. So you told that no performance will not decrease. So then my question is,
No,
if
like if
performance,
everything, if no, there is no present. No, my question is if there is no detrimental alleles, then why performance will going to decrease?
yes, sir. Okay. So my question is, some
Yeah.
plants will show additive gene action, and some plants will show, some traits will show additive gene action, and some traits will show dominant
Yeah.
gene action of a particular plant. So if performance is not
Yeah.
decreasing, it doesn't mean that we fix dominant gene action.
Yeah, but now see maybe like for example, if it is we're saying that it will if it is not fixed means it will segregate. Okay.
Sir.
See, you are telling you are yourself is telling. Wait. For example, sorry. See, take this one is your trait which is giving a dominant gene action. It is not fixable. You are telling. Then, although it is not fixable, but why it is performing good in the next generation? This is your question. This is your question.
Sir, my question is, sir, in books it is said that dominant gene
See,
action is
see,
can't be fixed. Only additive gene action can be fixed. So if it's
see. I
a
am telling.
uh,
See, this is capital. This is heterozygote. Normally, in heterozygote only, we'll have maximum dominant gene action. Okay. Normally, it's like that. It's
uh,
homozygous also so dominant gene action. That, but it's exception we will discuss later. But normally, if you take this is a heterozygote, it giving
Yes,
dominant
sir.
gene action, it is not
Yes, sir.
fixable for the next generation.
Yes, sir.
Then you are saying that if there is no determinantal of genes which are present in this particular inbred, the next
Yes,
generation
sir.
also it should
Yes, sir.
it is performing good means this particular dominant is not fixable, but why it is performing good? That is your question.
Yes, sir.
Yeah,
Or else the statement should be dominant gene action can't be fixed. Sorry,
yeah,
dominant gene action
yeah.
can be
That
fixed.
can be. No, no, no. Dominant cannot be fixed. But although why the superiority is maintained, that is your question. Even though dominant is not fixed, why the this is fixed? Like why it is showing superiority? That is your
Yes, sir.
question.
Yes,
Yeah.
sir.
And you are also saying there is no determinantal effect.
Yes, sir.
If small a is not showing determinantal effect, capital A is not showing determinantal effect. If capital A is not showing determinantal effect, then why the performance is going to reduce even after segregation? That is my point.
Huh. So that's why I'm telling that in some textbook they will say that
Yeah,
dominant gene action can't be fixed. Here in this
can't
situation
be
doesn't mean
fixed.
that dominant gene action is fixed.
See, the performance, see, normally what will happen? Normally in nature, if this particular A is there, if small a is there, then definitely there will be two different forms of the different trait. Okay.
Yes, sir.
So if capital A is there, tall means, then alternative allele means definitely there will be another phenotype. Many times.
Yes, sir. Yes, sir.
Okay. You want to select this particular trait which is governed by this particular allele. Means
Yes,
when
sir.
it will fixed, when it will be in homozygous condition.
Yes, sir.
But normally dominant gene action it's normally governed by heterozygous in nature.
Sir,
You
that is
can't
what I
fix
mean.
this.
Sir,
That is
actually,
normal.
if it's
That is not
sir,
over
if you say
dominant.
word dominant gene action can't be fixed, then it is right. But capital is the dominant gene action, right?
No,
It
no,
can
no.
show the
That
gene
can
action.
always heterozygote will segregate. It cannot be fixed. That is a simple genetics. If it is a heterozygote, what is fixable and what is non-fixable? First, you give me clarity.
1 second, can I interrupt Prem Kumar?
Yeah, yeah.
Pooja here.
Can yes, please.
Yeah, actually, I understood your question. You are wanting to say that dominance gene action, even like if
Yes.
capital A also is considered like I think you are little bit confused between dominance gene action and over dominance, because whenever we
Yes,
say
ma'am.
about uh, dominance gene action, we are comparing always the heterozygote with either of the homozygotes. Okay.
Yes, ma'am.
So to Jayant, I'm saying whether like
Yes, ma'am.
when we are saying talking about dominance or over dominance gene action, we are comparing the heterozygote only with the either of the homozygotes. Okay, so if
Yes, ma'am.
the heterozygote is performing better than both the homozygotes, then it is over dominance. And if the heterozygote is equivalent to that of performing equivalent of that of the what is that called? Dominant
Dominant
homozygote,
homozygote.
dominant homozygote, then it is called as dominance. Okay, so
Yes,
when
ma'am.
we say that well, like in case of the example that you said, like in inbreds, so
Yes, madam.
usually like we cannot generalize and say that inbreds are having additive gene action like that, because it is depending on trait. So each and
Yes,
every
madam.
trait will have a different kind of gene action. And in nature, it will not be possible even after inbreeding also that all the traits will be purely homozygote. And in case of mainly cross-pollinated crops and all, it is very difficult to attain such a situation in a natural population where all the traits will become completely homozygous, and all your detrimental alleles will be gone. So that is
Ma'am,
even
that's
that
why I
is a
told
very
hypothesis situation, madam.
It is so the situation that you are saying. In that case, the population will have all like the situation that you are saying. In that case, there will be no heterozygote in the population. The population will have only dominant homozygote genotypes in them.
Yes, madam.
Okay. So we don't like if let's say that for a particular trait, all the genotypes are having dominant like dominant homozygotes, then that trait has got fixed. You can't do anything to that trait. So that trait will not segregate and that trait will be fixed. But that since that trait is homozygote, that cannot be called as your dominance gene action. So that is your additive only. Okay.
Madam, okay. So my question is, can gene action will change in trait?
Gene action change in trait means
So
okay.
you're telling that before it was dominant gene action. Now after homozygote, you're saying that it is additive gene action. It doesn't mean that a gene action will change.
See, gene action will change. That is why gene action is changing means we are studying
Means
gene action
will it
usually
change gene
when
action?
it will change depending on the population and depending on the genotypes that you are studying. Otherwise, why we are doing gene action study in each and every population if we already know and if we already fixed like if it is fixed, if the gene action for it, let's say for example for rice yield, gene action is additive. If it is fixed, we know that. Then why we are going on studying for each and every like each and every MSc thesis, we do a different kind of gene action study. We do why we do is because the panel that we choose or with the population that we use, depending on that, depending on the environment and many things are there. So it is not straightforward. If it is like that, then we would have got one table in BD saying that for this trait, this is the gene action. It is fixed. You cannot like it is not change, but gene action will change. So that is why we have to study gene action when we are using a different set of panel of genotypes.
Thank you, ma'am. Really, it's very good insight. Thank you, ma'am.
Okay, madam.
I think your doubt
Madam,
is solved, no?
Ma'am, means
Little
I
bit
don't know
solved.
whether not little bit short because I don't know whether this gene action can be fixed or not. But
That is what
I
gene
still
action.
have doubt, madam.
See, that is what gene
No,
action will be
actually,
fixed and not fixed. Is theoretically what we study is different from what happens practically. Okay. So that is why gene action is continuously being studied by each and every person because it is not
Ma'am,
guaranteed.
ma'am, what I think means after means if all are dominant means all are homozygote, then additive effect might change. But shifting from dominant gene action to additive gene action, I think it's I'm not satisfied, madam, because even in Dharwad
see,
SS Padis are
actually,
told
can
that
I?
was asking same question.
Hmm.
So
No,
No,
actually,
that is what.
for one
See,
trait, yes,
yeah, you
ma'am.
tell.
No, actually, that the same point I'm going to tell you. Like in one, it
Huh.
depends. Like all your estimates of gene action, heritability, genetic variances, all these biometrics. It depends on sample size, depends on which like population, and depends on most of the like which pesticide you are going to analyze. Okay, for example, some trait you are getting additive gene action in particular population of the rice. If you are using another population of the rice only, in that particular trait may be showing dominant gene action in other population. That chances are there. It's not like you can't generalize a single, for example, plant height. It's saying in rice plant height is additive by studying your population. But if I study the another population, the plant height may be a follow dominant gene action. It happens. Now you understand. It is not like that you studied
And
in one rice population website. Yeah,
madam,
yes, ma'am.
sir, means what I think is
Yes.
so if we keep on going means homo means if we increase the homozygosity, the additive variance will increase, and
Yeah,
we can say
definitely.
that from dominance variance it is shifted from dominance variance to additive variance.
No,
But
it is
I
not
think
shifting. We are not saying it is shifting. See, for example,
shifting in the sense means it is going in the direct means additive variance is increasing and dominant variance is decreasing.
That you can say.
Yeah.
And another thing is like that situation that you gave, na? In
Yes,
that
madam.
situation, you are saying that like inbred, it is pure inbred. That what the situation what
Yes,
you are
madam.
saying.
Yes,
In
madam.
that case, it is additive gene action only, and in that case, it will not change. See, it cannot change from additive to dominance. No, somewhere from
Yes, madam.
dominance only it can come. Like if the entire population is having that, so we are, let us say, if you are taking F2 population, then you
Yes,
will
madam.
get all kinds of like gene action also.
Yes,
So
madam.
you cannot say that it is like that. But if you take an inbred population for that same trait, you will have additive gene action only.
Yes, madam.
So it depends on population and trait and the things. That's why we keep on studying that for each and every this thing.
Thank you,
Okay, madam.
ma'am. Sir. Thank you, ma'am. Really, it's a great
Yeah.
information. And madam is student of SS Patil, sir, and definitely madam is saying.
Madam, we are also students of SS Patil, sir.
Yeah, I
Dharwad.
did my MSc from Dharwad.
Oh, yes, madam.
So it's like very good
Now he's not teaching in
information.
Dharwad.
Okay.
Yeah.
Mhm.
Okay. Then like actually biometrics things, it's a very debatable things actually. Okay. Now you got some ideas. Then
Yes, sir. Yes, sir.
and thank you, ma'am. Thank you. It's really helpful. And madam will also
Thank you.
take classes on
Actually,
biometrics.
I'll see.
Yes, ma'am.
Actually, I have to
Yes.
take out some time for that.
Yeah.
So let me
Okay.
see. If possible, I'll tell you. And
Yes,
I
ma'am. Okay,
like
ma'am.
to have such kind of discussions. So that's why I keep
Yeah, ma'am.
joining. So
Actually,
if some discussion is there, we can
yeah,
get
ma'am.
some,
Actually,
because
even
breeding is little bit genetics is straightforward, but breeding we need more
yeah, ma'am.
discussions to get our doubts clarified.
Yeah, ma'am.
So
Actually, earlier people used to join in very large number, ma'am. Actually, we used to have a lot of discussion. But nowadays, nobody is there
uh,
and nobody asking questions, and just I'm keep on telling the points. And they'll not question also, ma'am. That's the problem. If the questions
mm,
will come, I will think, and either if I don't know answer, okay, I will search and I will come in the next class and I will tell. This is what,
yeah,
yeah.
that is what I see, that you are taking, like Prem Kumar is taking good efforts, but I hope
Thank you,
more
ma'am.
people will join and more
Yes,
discussions
ma'am.
will happen.
Yeah, ma'am, actually. I don't know why they joined, ma'am, paying the fee and they joined, but they're not attending the class. So many YouTube classes also available if they want recording. Okay. That's what,
Maybe
ma'am.
they just want to have the recordings and
Yeah, ma'am. That's the problem. Thank you, ma'am. Please join
anyhow.
for subsequent
Yeah,
classes.
yeah, okay. Thank you.
Yes, ma'am. Thank you, ma'am. Yeah. And I have a continuous class biomedical genetics, and madam also I think take the classes. Okay, you got all the points. Okay, shall I close?
Okay,
Okay,
sir.
sir.
Okay, good night, everyone. Bye.
Good night, sir.